want to thank the Society very much for allowing me to give this plenary. I've never given a plenary at Waterbridge before, and it's a real honor. You know, as ecologists, we are constantly tasked with this idea of trying to understand relationships between biotic and abiotic worlds. And the problem is that our biotic components are almost always adapted to certain conditions. And the anthropogenic effects are almost always outside those conditions, or very often. So what you end up with is a very muddied answer, almost always. And it's very, very difficult to disentangle what's going on. And especially in that context, to make recommendations for conservation or management. The story I'm going to tell you today is kind of a rare one, where we were able to spend 35 years studying something that really allowed us, I think, in the end, to do some real disentangling and really understand where the anthropogenic leaves off, where the natural biotic is probably in sync, and ultimately how to manage this really large wetland ecosystem. So to start off with, I just want to give a little bit of background on the Everglades. Its drainage is the size of the state of New Jersey. It's in the southern portion of the Florida Peninsula. It is an extremely flat landscape where water doesn't really flow. If you want to mimic the Everglades, the easiest thing to do is take a small pitcher of water and slowly pour it out on a flat table. The water simply spreads out. It doesn't get any deeper. And if there's any pitch to the table whatsoever, it flows very gently in that direction. In the Everglades, that is exaggerated because it's such large distances, 200 kilometers from top to bottom. And it takes so long for water to get from one end to the other that the water may evapotranspire and rain back down 3 or 4 times. So that gives you a vague idea of how water moves and what this landscape is really like. This is a hypervariable hydrological system. We have a wet, dry monsoonal cycle in which we get most of our rainfall, 80%, between May and November. And then the rest of the time, it's mostly pretty dry. In addition to that seasonal cycle, we also have this very different rainfall from year to year, largely due to tropical systems that may move in and dump 80% of the water in a year in a day. We also are very susceptible to things like El Nino and North Atlantic oscillation. And so we have a really chaotic system dumping water on a very flat landscape that magnifies that hydrology. Another fascinating thing about the Everglades is that it has extremely low phosphorus inputs. The water in the Everglades has less phosphorus than the rain falling in it. And I can go into that in questions if you like. The biomass is dominated by small fishes for a couple of reasons. So first, the productivity is generally very low on an aerial basis. And secondly, because of the wetting and the drying so frequently, the big fish don't really get much of a chance. And so most of the biomass of fishes, at least, is less than 7 centimeters. This system in many ways is tailor-made for wading birds. So the wet-dry cycle is one that causes the prey to become very concentrated. And so in the top and the bottom of the ibis pictorial here, you've got just as many fish. It's just that they're forced out of the vegetation and into little pools where the birds can get them through a variety of foraging tactics. So this means that you can take a system that is essentially oligotrophic and unproductive and turn it into one that puts on one of the biggest aquatic bird shows in the world every year. I began working in the Everglades in the late 1980s, and we already knew that a lot of things were wrong. I'll go into that in a minute. But basically, we were trying to figure out what drove wading bird nesting. Is it food? Is it predation? Is it disease? Is it something else? And I spent the first 5 years trying to figure out, or essentially proving what didn't drive wading bird nesting. And the amount of nesting substrate is very abundant. There is very little human disturbance. Disease turns out not to be a big factor. And predation, nest predation, also does not appear to be a big factor. We're left with food and food availability as the really big issue in the Everglades. The picture that I'm showing here is of a 1941 colony, Broad River. I'm going to come back to that colony later, but this was essentially during a period that produced a lot of wading birds and supported a lot of wading bird nesting, primarily in the coastal zone. The low predation has an interesting backstory. It turns out that there's very little predation because alligators are patrolling these colonies of birds and essentially protecting the wading birds from raccoons. The story is that there's actually a symbiosis there, that the wading birds are actually attracted to alligators, apparently because of this protection, but the alligators are attracted to the birds because there's lots of nestlings, in some years, hundreds, falling into the water which the alligators eat. So all this is bad news for the raccoons and good news for the wading birds. All of the alligator decoys you see on the lower right were part of a massive experiment in which we demonstrated the actual attraction. Lucas Nell, who was a master's student some years back, demonstrated that 40% of the female population of alligators in the Everglades can be supported for 6 months just based on nestlings. Of course, we have dramatically modified the Everglades, particularly in water flow. And in many cases, people call this the largest water control project in the world. We have moved most of that flow from going into the Everglades. We've now moved it to going out the 2 outlets from Lake Okeechobee. And those are massive dumping to sea of fresh water. The reason is because we were trying in the 1950s and through the present to maintain a large agricultural system immediately south of the lake, and we didn't want to flood it. It also helped that we compartmentalized the Everglades into a series of impoundments so that we could control flooding. The net result, though, was that the Everglades itself, and particularly the southwest coast where the estuary is, were grossly dewatered. And so that has led to a lot of really nasty consequences. The little panel in the lower right, it demonstrates the, I didn't put the axes on here, but it's basically the x-axis is days since drying, and the y-axis is the relative density of bluefin killifish in this case. What you see is a bell-shaped curve. So at since the drying has just occurred, you get very few killifish. And then as time since drying proceeds, it increases and then decreases at the top end. It decreases at the top end probably because the large fish are able to grow and come in and chew on the killifish. But all of the small fishes have this sort of sweet spot relative to hydroperiod, or in this case, days since drying. The left panel shows some work by Bill Loftus, which is throw trapping data on small fishes in the Everglades, and it's over a long period of time. You can see the trace of the wet seasons, which are the low densities when fish are not concentrated, and the peaks, which are the dry season when fish are. And you can see it really varies a lot from year to year. It is, as I say, hypervariable. The interesting part, though, is to look at I don't know if you can see my cursor, but these lower darker dots are another system that he was studying only a couple miles distant that were highly dewatered. What this indicates is that by reducing the amount of time that water sits on the land, you are grossly reducing the amount of time that fish have to grow, and therefore the standing stocks of those fish. The consequences on birds were already well known by the 1980s, and 2 of my mentors, Bill Robertson and John Ogden, had demonstrated a couple of really dramatic effects. The first was that, at least by comparison with the 1930s and 1940s, prior to ditching and diking, we had a huge reduction in numbers of nests, 80 to 90% reduction. It's still variable from year to year, but boy, it really took the ceiling out. The colonies also moved away from the coast. Remember, the coast is the most dewatered part of this system, and they moved inland, albeit in much smaller numbers. John Ogden demonstrated that storks had begun nesting later and later over time. And the reason was because they were finding less and less food around because standing stocks were low. So they had to wait later in the season for increased drying to occur before they could get enough to start nesting. That led to reduced nesting success because they would start so late in the season that they didn't have time to bring off a brood before the beginning of the rainy season when the prey dispersed. So pretty devastating effects on birds. To make a long story really short, this has resulted in a major hypothesis for restoring the Everglades, and in fact underlies the currently active comprehensive Everglades restoration program, which is moving dirt around and doing some great things. The idea is that if you increase the flow, particularly to the estuary, but throughout the Everglades, you're going to get more prey fishes, and we're pretty sure of that, and that they are going to be more available to birds. And that will lead to increased wading bird nesting, particularly in the estuary. I believe firmly in this hypothesis, and I think it's really one of the major keys to the Everglades. But there was something that was really bothering me since the early 1990s, that we have also seen a huge increase in the amount of mercury burdens in all of the top predators of the Everglades. So raccoons, great blue herons, Florida panthers. We had a Florida panther die acutely of mercury toxicosis. That takes a lot of mercury to do. This was a really big surprise in the early 1990s, and nobody knew where it was coming from. The Everglades was thought to be pretty clean of contaminants, and so what really was going on? Well, again, to make a very long story really condensed, a lot of that mercury was coming from the urban East Coast. The urban East Coast has essentially no place to put its trash, and so the vast majority of it is burnt. So landfills, the water surface is right below the surface of the soil, so landfills don't really work. And besides, it's mostly urban, so it's hard to find a place. As it turns out, most of that, the effluent from those incinerators goes back out over the Everglades. For much of the year, the prevailing winds are southeast. And because we have such frequent rainfall and thunderstorms in the summer, most of it gets rained back out as wet deposition onto the Everglades. Really interesting part of this story is that a huge portion of that mercury was actually coming from two things. One was medical waste incineration. That was a small portion, but a huge portion of it was coming from small household batteries, which at the time had mercury in their formulation. And because we went through so many of them and put so many into the incinerators, that was probably 70% of the mercury going into the Everglades. As it turns out, the Everglades is an awful place to put mercury. First, in a terrestrial or a lacustrine system, much of that mercury would end up in the bottom sediments, and it would end up being buried over time. Because this is a wetland that dries out and wets on a fairly regular basis, it doesn't get a chance to do that. All those sediments are oxidized during the dry, and then they become more available to biota with each cycle. The second problem, and this was a real shocker, is that most of it was being methylated. So methyl mercury is about 10 times more toxic than elemental mercury. And methylmercury is indeed what travels in food webs, and it's what you're worried about in sushi grade tuna, and it's the real problem. It crosses the blood-brain barrier. Methyl mercury is the real problem. In the Everglades, it turns out to be methylated extremely rapidly. Up until people had studied the Everglades, high rates were from Lake Erie, where 40% of the mercury might get methylated in a year. In the Everglades, they found 100% was being methylated overnight. And so it turns out we just have really good conditions, pH, sulfate levels, to be able to do that. The last problem with the Everglades is that we have this enormous biological accumulation factor. And this is because the food webs are complex and long, and a lot of things eat a lot of things. So between water and a largemouth bass, the accumulation factor is about 1 million times. With wading birds, it's more like 3 million times. So all these things together, the regular reliberation of mercury, the very high methylation rates, and the biological accumulation factors make mercury go, a little bit of mercury go a long way in the Everglades. Now, over time, mercury has actually decreased very markedly in the Everglades, or at least the local human inputs of mercury. So this is a graph over the last, 80 years or so. And you see this really big reduction in the early 1990s. The red in this graph is those household batteries. And the yellow in this graph is latex paint. I didn't know for a long time that latex paint actually had mercury. Both of those were taken out either by the industry or by regulation. And so by the mid to late 1990s, we were having inputs to the Everglades that were very similar to prior to World War II. This was an enormous reduction, one that got almost no attention, and yet was an enormous unplanned ecological experiment over a very large ecosystem. We'd been tracking mercury in the feathers of young great egrets, because young great egrets are getting their food primarily from the surrounding landscape, and they have a relatively small contribution from the egg. So they seemed like a pretty good metric, and it was geographic. Great egrets nest all over the Everglades, so we could look at that. Each one of these lines here is a colony tracked through time, and obviously some colonies don't last all the time or are not active in every year. But the really big story was that we saw a big reduction around 1998. And some models suggest that that is about how long it might take for the reduction in mercury source to work its way through the ecosystem. So we had a big reduction, at least 65% during that time. So remember 1998? About that time, we also had a really big increase in the numbers of nesting pairs. So between 3 and 5 times increase in white ibises, great egrets, and wood storks. This is pretty hard for a biologist to ignore. This is exactly what we expect from restoration, but we hadn't done any restoration. So, or at least hydrological restoration. So this was a bit of a mystery. We called it the bump, and we began to wonder if the release from mercury might have had something to do with that increase. Of course, this is messy. It's pure correlation, and really this is just throwing spaghetti at the wall, because we don't have any information on a lot of the other things that might affect this, like food availability. But it did spur us to try and understand what was happening with mercury a little bit better, and how powerful it might be for nesting and wading birds. Mercury is a real problem because it affects multiple physiological systems, and that's it makes it actually fairly hard to study. I want to distinguish between sublethal and lethal mercury exposure. Of course, we don't believe that mercury was high enough for birds to be dying, at least not directly. But at least on reproduction, there's a lot of different routes that might affect behavior, might affect your ability to hunt, it therefore might affect survival and the ability, your parental abilities. There's also direct effects on mortality of embryos. We know that from lots of studies. And altered hormone expression is one that really rose to the surface, that mercury has increasingly been pegged as an endocrine disruptor, and that could affect both the thresholds to abandonment once you've started nesting and the thresholds to breeding. It also can strongly affect parental abilities, courtship behavior, and so forth. So that's kind of where we began. The first studies were led by Marilyn Spalding. We raised some great egrets in captivity from egg, and raised them through the fledging period. And although none of these birds died, they had plenty of food and not many stressors, they all showed some pretty strong effects. And these are at levels of mercury that you would see in the Everglades. This is not a typical toxicological experiment where you start off way beyond that level and then back off. We always went for environmentally relevant doses. So these birds had reduced pack cell volume. They didn't have as many red blood cells. Their lymphoid tissue was reduced. Their appetite, motivation to hunt, they were lethargic, didn't eat much. They lost weight, of course. And they had evidence of decreased immune function. Now, none of these birds died, like I say, but if they had been fledging into a typical natural system, they might have had a hard time of it. Julie Heath was a former PhD student in my lab and has been at Boise State for many years now. In her dissertation, she demonstrated that both in the field with white ibises and in the lab with zebra finches, that levels of mercury that you might get in the Everglades really do play with the hormones that control reproduction: estradiol, testosterone, and progesterone. In the case of zebra finches, she was actually able to demonstrate much lower hatching success and lower reproductive success overall. So that really put us on the road to, wow, there's a mechanism here that might be powerful enough to affect birds at the population level in the Everglades. And this caused me to go on this sort of crazy ride with an aviary where, at this time we were really, Everglades restoration was really ramping up. The hydrological restoration hypothesis was well developed, and it looked like that train was really leaving the station. And I was deeply concerned that we might have restored the Everglades hydrologically, but still have a major mercury problem that was essentially a confounding factor. So for this reason, we began we raised these white ibises in captivity on Everglades relevant doses and raised them to the point that they were capable of breeding. So we really wanted to know what that actual breeding effect was, and this appeared to be the only way. We were first able, and this was a bit of a surprise, to mimic the levels of mercury in the birds in the Everglades. And so the low, medium, and high mimicked the control low, medium, and high. This is where they stand in this graph of historical mercury. The pre-1980s was low, and then high was in the 1980s and 1990s. So I was very relieved about this. This was a double-blind experiment. Somebody might have put the wrong food in the wrong place, but evidently we didn't. Nilmini Jayasena led most of these aviary studies. The first thing she found was that the dosed groups had fewer nests with eggs, and that was largely because of a lot of pairing between males. So 55% of the males in the high dose group were pairing with males. She also found that male-male pairing was dose dependent, and that dose males had very strongly altered hormone expression. Meanwhile, the heterosexual pairs also had a lot of trouble raising young, and I'll come back to that in just a minute. Nilmini, I should also say, is now at the University of Peradeniya in Sri Lanka, and she's continued to collaborate on this work. The male-male pairing was really odd. We didn't expect it at all. These males had durable pair bonds. The whole show included everything you would see: courtship, nest building, mate defense, but no eggs, of course. This wasn't because there were too few females. The sex ratios were even or female biased. The homosexual males were fertile. In many cases, they went on much later to pair with females and produced fertile offspring. The females responded a lot less to homosexual male courtship, and there are two basic displays that males are doing. There's a snap stretch and a bowing display, and then an acceptance display. And it turns out the mercury birds were doing this a lot less well. So in this graph, 0 is essentially controls. And these low, medium, and high are the low, medium, and high groups, and then the homosexual birds, which were distributed between the medium and high groups. Their bowing and acceptance rates were much lower than the controls, all of them. It did appear to be dose dependent, and the homosexual birds were the ones that had the lowest bowing and acceptance rates. So the story that emerges is not that mercury makes you gay. The story that emerges is that males are impaired by this mercury, and it is really messing with their hormones, and they're unable to do their full courtship display because they don't have enough testosterone. So I think that was a real eye-opener for us as to the mechanisms of how this might lead to fewer pairs. So in the end, we saw compared to controls, we saw about an 18% reduction in nest success, because of that homosexual pairing. And then we saw a 35% reduction due to poor parenting by heterosexual pairs. So the heterosexual pairs would look at their eggs, incubate them for a while, stand up, and then drop one of their eggs onto the ground and watch it. They would feed their young and then turn around and really aggressively beat the young up to the point that their heads were bloody. So some real aberrant behavior. All of the mercury work in the aviary led us to this basic idea that sublethal mercury exposure was leading to endocrine disruption, which was impairing reproduction to the extent that we're probably seeing population level effects in the Everglades, or at least that was the idea. However, it is notoriously difficult to translate stuff in the lab to stuff in the field, because you've got all these other stressors and sort of biological washout and things going on that are really hard to control. So at this point, we really just had that lab study. Fortunately, we also had been accumulating a data set over time that was reproductive success and mercury exposure of great egrets over about 20 or 35 years, depending upon the data set. And this was in colonies in the Everglades. And so we had something to work with. This is a messy data set because, of course, it does include all of those nasty stressors that you can't control for. So you might know that mercury is increasing or decreasing and reproductive success is doing the same, but you don't know why. Fortunately, one of my close colleagues, Joel Trexler, who's now at Florida State, had been doing for many years an assessment of fishes in the Everglades. And so this was trying to figure out what standing stocks and densities were like. And he did it all over the Everglades. This is a huge amount of work. And if you've ever worked a throw trap, you can spend about 5 minutes at one of these and say, okay, I'm done. It's not much fun. So really heroic data set. What it meant was that we could look at specific colonies in the Everglades and know in any year what the standing stocks and densities of fishes were like. That meant that we could control for food while looking at the local and annual effect of mercury. So our idea becomes something like this, that food availability may be working through body condition to affect reproductive impairment and also interact with the effects of mercury. An animal in higher body condition might be better able to withstand the injury caused by mercury. This work, this analysis was led by Javi Zabala, who's a postdoc who's been in my lab for about 3 years and has done some really masterful work. So the first thing that we did was we began modeling hatching success in relation to both mercury and of food availability. So we did this by looking at both fish standing stocks and how available those stocks were becoming to birds through water recession. And the variable of interest was the nest-specific hatching rate. We wanted to control for colony and year effects as well. When you look at this, all these dots here, of course, are different colonies in different years. When you look at this hatching success in relation to mercury, that's colony specific, you don't get much of an effect. But this is when you're not including the effects of food. So it looks like it's just a bunch of unrelated data. If you do include food effects, you get a very different picture. So here the thin line is the one that I just showed you. At high food availability with lots of fish, you also get almost no effect of mercury on hatching success. So it appears that the birds are able to compensate for whatever injury is being done by being in good nutritional condition. But if you have not very many fish or they're poorly available, you get a really different story. So at the exact same mercury level, you can pick 10 or 15 parts per million in feathers. At the exact same mercury level, you're getting a really different response depending upon how much food is available. This is interesting. This means that nutrition has a really big effect on how the contaminant is affecting animals. This is an idea that is only just now beginning to circulate in the human literature, and this is one of the the first real big ones for wildlife. So we do have effects. They are strongly mediated by food availability. Hobby was also interested in nesting propensity, or the probability of getting to the egg stage, because if your hormones are being messed up, like the ibises, you might not even nest at all. So his model here was pretty much the same. It was at the colony level, and it included all the same effects. He also included some rainfall, wind, and temperature, because those are big annual effects. And we're interested in numbers of nest initiations. In this case, we really were interested in this local colony effect, because big colonies and little colonies have more birds in them, right? What we're really interested in is how those vary from year to year. So that random effect, that deviation from normal, was really what was driving this, this analysis. So again, a really big effect, over the range of mercury that you might see in the Everglades in feathers. You get a 50% reduction in numbers of nesting pairs. And that's while controlling for the effect of hydrology. This was a strong model. So the effect of all of the main, the main effects, the main variables, was explaining about 70% of the variation. If you looked at the whole model with the random effects, it was explaining about 99% of the variation, which was really obvious evidence that Hobby in the dark of night had made a pact with the devil, because you never get models that explain that much variation. So in any case, that was very surprising. Hobby also went back to Nalmini's data and analyzed the ibis data and looked at pretty much the same thing, the propensity to breed. The effect is less, about 25% reduction over the range of mercury, but it is nonetheless pretty pronounced. This suggests two things. The first is that our mechanism in the captive white ibis, where they were experimentally dosed, is actually mercury. And the second is that there may be a pretty strong difference between lab and field because of those field stressors like food. What this suggests is that we ought to be really careful about applying lab results to the field. And I'll get back to that in just a moment. Okay, Hobby also was interested in going back to the bump, that big increase in numbers of birds. Here I'm showing not all the birds, but just the great egrets, which is our best data set and the one that we've modeled. These are the actual data. That's 1998. This is the before and after mean plus one standard error. There are the means of those data sets. Hobby went back with the data, with the model that was developed during the pink period, and tried to forecast what would have happened during the blue period, given the amounts of mercury that were there. His average was pretty much spot on, suggesting that our hindcasting abilities are pretty good. So this was, in a way, one validation at least of the model, and it certainly suggests that mercury is having a pretty big effect on nesting numbers. So Hobby also went on and put together these really cool graphs that try to both integrate all the things that are going on, like probability of breeding and also failure of breeding, nesting success, but he also assigns it to nestling death, hatching failure, and so on. So the white portion of each of these graphs is the number of nests you would expect to be successful over the range of feather mercury. And by successful nests, I mean they're producing some offspring. So that's the scale on the left. So at low mercury, they would be expected to produce 1.2 offspring. And of course, some of them die and some of them don't hatch. And you might have some pre-breeding failure. But as mercury increases, you get a really big increase in that propensity to nest. And after some threshold, you get hatching failure really coming in as a major term. Okay, this is at median food availability over all the years that we looked. If you look at high food availability, so this is where you have big recessions and lots of fish around, you get a lot more successfulness, and that expected offspring per adult is a lot higher in those nests. Pre-breeding failure is still a big term as you increase, but hatching failure, we apparently are not hitting a threshold at high food availability for having that hatching failure. Nestling death, which is a fairly normal process, which is mostly staying constant in these, becomes relatively larger at that high food availability. Okay, what about at low food availability? There we end up with a lot fewer successful nests, and even at low mercury, they're producing only 0.8 offspring per adult. Hatching failure, we hit that threshold a lot sooner, and pre-breeding failure is a really big term almost throughout the range of mercury. So this gives you a pretty good idea of exactly how mercury is affecting these things in relation to food. So to summarize this work, mercury, I think we can say pretty safely, is a very strong contributor to population change at the level of an entire ecosystem. We are forecasting 60% reductions over the range of mercury that's out there, and that's combining both nest success and pre-breeding failure. So this is a pretty major effect at sublethal levels of mercury. A lot of the effect is on breeding propensity. So 50% reductions over the range of mercury. It is probably occurring based on what we've seen in the lab through endocrine disruption. That is probably the main mode of action. Those mercury effects are really strongly modulated by food availability, probably through nutrition. A lot of the effect on breeding propensity and on breeding success may be because animals not only are affected by their hormones, but they also just don't have the nutrition levels to make a go of it. This is a really strong finding, and one that has a lot of implications for ecotoxicology around the world and lots of different contaminants. We need to start really looking at the ecological factors that are affecting birds in addition to the contaminant if we're going to understand the effects of the contaminant. It's almost impossible to disentangle them in this case. And lastly, just lab studies are quite likely to underestimate the effects of mercury. This is not a particularly new idea, but it is one that I think we've demonstrated sort of the magnitude of that effect. Okay, so back to the Everglades. We are in the middle of an historic restoration in the Everglades, where we are going to be shunting water away from the two canals on the east and west of Okeechobee and putting a lot more into the Everglades itself. The goal is 1.5 million acre feet. That's an acre of land 1 foot deep. It's a huge amount of water. We may get about 750,000 out of the current restoration. We're not sure how far that's going to go, but in any case, it's probably a really strong thing for the Everglades, and I believe all our predictions are likely to be pretty true about the fish and the birds. So this is all very positive. But what does our information about mercury tell us about this? Well, the first thing is that we're going to get a lot more water, a lot more fish, and a lot more birds eating those fish. This is some work by James Behrens, who's with USGS, and Dale Goblik at Florida Atlantic University. And this was some modeling that they did that is comparing the restored and unrestored conditions. And basically all the orange areas are areas where we're going to have more water. And that's where you see a lot more fish production and a lot more birds. The interesting thing is that if you do this over an annual cycle using typical cycles of water, you end up with 2 to 3.5 times more birds net foraging in the Everglades. That's a lot of birds. What this suggests is that we're likely to have a lot more well-fed birds. Based on the information that we've been doing our research on, this suggests that we're going to have a lot fewer problems with mercury at almost any level. So I think that's actually really hopeful. It's almost impossible to totally disentangle an effect of hydrology from an effect of mercury, but both of them are going in the right direction. Okay, I started this talk about you know, what does an unaffected ecosystem look like? What is one that is unmuddied by anthropogenic effects look like? How do we know when we sort of got it? In 2018, we got a really unusual year. We got a year in which we had really massive rainfall, far out of the ordinary. It wetted the whole Everglades. And in the end, it looks like the amount of water that we got really mimicked what we used to get pre-drainage. We had a long growth season for the fish, and then a really nice dry down. We had a low mercury year. And so this is actually what that looks like when you, sorry, this is a video that I don't seem to be able to play. But in any case, this is what it looks like, is really big colonies all over the Everglades. We had the largest nesting event since 1940 in 2018. We had about 120,000 nesting pairs of all species. In most years, we get 10 to 20,000 nesting pairs. So this was an enormous increase. And what it demonstrated clearly is that if you do get the water right and you do get the mercury right, you are likely to get a really massive increase in the numbers of birds. The smaller, highly controlled Everglades is still capable of supporting enormous populations of birds. I just want to close by saying that, you know, this has taken a long time to understand, 35 years and at least as many researchers. We all stand on the backs of those who are in the black and white, who are mostly now deceased, but who set a lot of the population levels and also told us about what this system was supposed to look like before we mucked it up. I think the answers here are rare, and I think that we were very lucky to be able to keep this ball rolling as long as we have. It shows us very clearly that our expectations for research, especially ecological research, ought to be for long-term research, multifaceted research, and stuff that really goes in depth after specific uncertainties and questions.